Howdy, folks. Thanks for checking in to MrUlrichLandofBiology.com. I am Mr. Ulrich. In this notescast, we're going to be taking our first tour through the cell, taking a look mostly at the endomembrane system, so the membranes that make up the cells and the structures that are bound in those membranes. Very recently, we've been discussing different types of cells, the nature of a prokaryotic bacterial cell, and what typifies that when we compare that to eukaryotic animal cells or eukaryotic plant cells. Of course, the big thing is going to be these membrane-bound organelles. Pro rhymes with no nucleus. Prokaryotes don't have a nucleus. They do have a nucleoid, so they have a region for the DNA. All cells have DNA. They have a region for the DNA to hang out, but it's not bound within a nuclear envelope, within a membrane, the way that it is in eukaryotic animal cells or plant cells or funguses or protists. Can't forget them. Big thing, of course, with them is that they have organelles. Though prokaryotes really do rule the world numbers-wise, eukaryotes are still doing quite well. What is the bonus? What is the benefit of having organelles in the first place? Well, for one thing, organelles are specialized structures. So things like cilia and flagella are attached with the cytoskeleton. So these are groups of proteins. We'll talk more about cytoskeleton in further videos that allow the cell to move, the organism to move, or things to move around the organism itself. One of the big bonuses, though, of having the membrane-bound structures is it allows for partitioning, compartmentalization in the cell itself, so that the cell has regions that'll have different environments. So you can have different pHs, different concentrations of not just hydrogen ions, but of enzymes and substrates and all different environs, characteristics that are needed for reactions to take place. And some of these are going to allow some enzymes to function in some parts of the cell that they won't be able to function in other parts of the cell, and that does protect the cell from the action of those enzymes. Things like in the lysosome, these enzymes are designed to break down parts of the cell, and so they need to be kept kind of sequestered to the side where they're not going to, they don't play nice, where they don't damage the cell and its contents. These are also going to serve as places for some enzymes to embed and allow for chemical reactions to take place, putting lipids and proteins together. So we start getting into lipoproteins and glycoproteins. We'll talk more about these when we get into the mitochondria. That's going to be in the next part of the cell tour when we start getting into energetics. All cells, whether they're prokaryotic or eukaryotic, have some basic jobs that they're going to have to take care of. For one thing, they all have to make proteins. Proteins are doing a lot of the work of the cell, enzymes, all that good stuff. And so they have to have some sort of machinery that's going to churn out the proteins necessary for metabolism. They also need to have some way of transferring energy from one type of molecule to another type of molecule so that they have the energy to run metabolic functions on a regular basis. And so they also have energy to synthesize new molecules and to grow. Speaking of growth, us multicellular organisms, and in order for us to grow, we have to make new cells. And we also fall down and go boom, so we got to repair those cells and replace them when they get old and gnarly. Really early on, we're taught the importance of DNA, and it is important, but it's not the workhorse. It's not what's really making metabolism function. All right, what DNA codes for are proteins, and it's proteins that are really the workhorse of metabolism. They're the things that hold the cell together and really make the cell work. And it's cells working together that are going to create these crazy organisms that we know and love and irritate us. Let's get a little bit more detailed here with the different functions of the cell. We'll start with building proteins. This really is the root of molecular genetics, is building proteins and how DNA is actually read and processed into functional proteins. In order to do this, like I said, we're going to spend a lot of time. We're going to spend a lot of time talking about molecular genetics and how those proteins, we've already talked about shape and the importance of folding, but really how we get to those shapes and how we actually go from the strings of bases to the strings of amino acids in the right order, and even adding those to other compounds to make glycoproteins or lipoproteins. It's dizzying, and we're going to spend some more time on that later on. But all cells have to have machinery for all of these stages of protein construction. And not only that, they have to get to where they need to go within the cell, whether they're going to be part of the cell or even secreted outside of the cell. So what are some of these organelles that are involved? Well, the nucleus is where the DNA is going to be stored. The ribosomes are the structures where the code of DNA is actually translated into amino acid sequences. Within the endoplasmic reticulum, which is a sequence of folded membranes associated with the nucleus, some proteins are made, more folding is going to take place, more modification and labeling for transport to maybe the Golgi apparatus, depending on what has to be done to that protein, so that it can then go into transport vesicles and eventually make its way out of the cell. So you start off in the nucleus, go to the ribosome, go to the endoplasmic reticulum, go to the Golgi apparatus, into transport vesicles, and then out of the cell if it's going to be secreted. This is what we call the endomembrane system, and we're going to talk about this at length through this video. Let's start with the nucleus. The job, again, of the nucleus is to protect the DNA. In the cytoplasm, there are enzymes whose job it is to digest polynucleotides, nucleic acids, down into individual nucleotides. So DNA wouldn't last too long out there. So good thing that we keep ours in a little bag. To protect it from the action of those enzymes. The bag is actually, we call it the nuclear envelope, and it's double layer. It's actually 2 layers of membrane, but there are places where the membrane comes together and actually adheres to create these little areas called pores. That's a bit of a misnomer. They're not really holes. They're more like gates that are made out of groups of proteins that kind of clog up that hole. They're big enough for really large molecules to pass through. Let's think. What molecules are going to need to leave the nucleus? In the nucleus, the DNA, remember, is read into messenger RNA through transcription, and it's that messenger RNA that then needs to leave the nucleus and get into the cytoplasm, where it's going to complex with the ribosome and make that polypeptide. Of course, remember, there's enzymes out there that are going to break that down. Those enzymes are important. We'll talk about that when we get into genetics. To show you the actual pictures themselves, you can see here there's the surface of the nuclear envelope itself. In this picture here, we also have, if you look even closer, getting really small, 1/4 of a micron. These are the pore complexes themselves. And those are the gaps where the DNA where the RNA is going to leave. This is a side view right here, and you can see the double membrane there. Within the nucleus are a small structure or structures called the nucleolus or nucleoli, if we're talking about more than one. Within the nucleoli is where ribosomal RNA is made. These ribosomal RNA molecules will complex with proteins in the cytoplasm. Of course, in order to do this, they're going to have to get out of the nucleus. So they're going to go through those nuclear pores, get out into the cytoplasm where they find the ribosomal proteins and form functional ribosomes. Each ribosome has 2 parts. It has the larger subunit and the smaller subunit, but each one is made out of ribosomal RNA and some proteins. Ribosomes are the site of translation. Their job is to take the base sequence of messenger RNA now and translate that into the amino acid sequence that will eventually become the protein. So the overall job of the ribosome is protein production. Their structure is there are two subunits. There's the larger one and the smaller subunit. Both of them are made out of a different type of RNA called ribosomal RNA or rRNA and some other proteins. When those two subunits combine, they make a complete ribosomal unit. Something to note, of course, we're not talking about any membranes. These are not membrane-bound organelles, so we're going to find them in the prokaryotes as well. Ribosomes are in all cells. All cells need to have machinery to make DNA into proteins. There are a couple of different categories of ribosomes. There are ribosomes that are kind of free and floating around in the cytosol itself. These ribosomes are involved in making proteins that are going to stay within the cell itself. There are also ribosomes that are attached to the endoplasmic reticulum that we're going to talk about here in a couple moments. The endoplasmic reticulum that has the ribosomes is called rough endoplasmic reticulum. It's the ribosomes that make them kind of look like they're more rough. But this is where proteins that are going to be destined for export or at least be part of the membrane or the endomembrane system are going to be built. Speaking of endoplasmic reticulum, though the ribosomes on the endoplasmic reticulum, rough endoplasmic reticulum anyway, though those ribosomes are where the proteins are going to be made, it's within the compartment of the endoplasmic reticulum where the proteins are going to be folded and get their proper shape and be combined with other compounds to make lipoproteins and glycoproteins. These are also going to be the proteins that are involved in the membrane itself, so the integral proteins and the signaling proteins, all the different proteins that are involved in the cell membrane, as well as other parts of the endomembrane system are going to be manufactured here in the endoplasmic reticulum. This is where other compounds are made as well. So the membrane-embedded enzymes in the endoplasmic reticulum do allow for the synthesis and hydrolysis of other compounds as well. The way it looks like kind of like a system of like a maze. It's a bunch of folded membranes back and forth. And again, what this does is create kind of compartments where there are different environments. There's different amounts of enzymes and the substrates that they need to work on in order to create these different important biological compounds. There are two different types of endoplasmic reticulum. There's rough endoplasmic reticulum. This is the one that has the fixed ribosomes associated with it, and smooth endoplasmic reticulum, which does not have ribosomes associated with it. The compounds that are created within the smooth endoplasmic reticulum are usually destined for incorporation into either the cell membrane or parts of the endomembrane system. In order to make these compounds, there's going to be a lot of different metabolic processes going to be taking place here. Some of them are synthetic. So since we're making new compounds for not just the membranes, but for other things as well, this is where quite a bit of lipid synthesis is taking place. So the phospholipids for the membrane system, as well as some of the steroids, which will be incorporated into the cell membrane, as well as become important precursors for things like sex hormones and other hormones. This is also where hydrolysis, where the breakdown of some of these compounds is going to take place. Things like glycogen get broken down into glucose within the cells of the liver. And also the detoxification of drugs and other toxic compounds is going to take place within the smooth endoplasmic reticulum within the liver. So this is where alcohol, as well as other toxic compounds, are detoxified. So the Endoplasmic reticulum is an important part of the endomembrane system. It does build the new membranes, and it does this by synthesizing those phospholipids and then incorporating those phospholipids basically into itself. So as the endoplasmic reticulum is making these phospholipids, it's going to start growing, and as it grows, it kind of bubbles off, it kind of blebs off into these, here's one right here, into these little bubbles, these little transport vesicles, little tiny things here like this, and then they're going to join, because remember these phospholipids are functioning much like soap bubbles do, and so they can kind of like join together into a larger bubble, we'll say, down here at the Golgi apparatus, or just become part of other parts of the cell that need more membrane and allows them to grow. Rough endoplasmic reticulum, kind of the rock star, kind of gets more treatment than the smooth endoplasmic reticulum, and that's because the rough endoplasmic reticulum has all of the ribosomes on it. Ribosomes, which, well, they're important. They make all of the proteins that are driving our metabolism, so kind of important. But the rough endoplasmic reticulum is really involved in making the proteins that are destined for export or are going to be part of the cell membrane, which are certainly important as well. So what types of cells are we going to see that have a preponderance of rough endoplasmic reticulum? Well, we're going to have to think of cells that are producing lots of protein. So things like mucus cells within the mucus lining that are producing lots of mucus, or hair follicles, or nail beds where we're making lots of proteins that are then secreted outside of the cell. All right, they do this by building the proteins right into those bubbles that I was talking about that are coming off of the smooth endoplasmic reticulum. Well, they come off the rough endoplasmic reticulum as well, and they are going to contain those proteins that are destined for secretion. And it happens here, let me get my pointer again, it happens here in the endomembrane system. So when we get a little bleb here of transport vesicle taking place, right? That's going to join here what we call the cis side of the Golgi apparatus. I'm going to talk about Golgi here in a second. Here it's going to be further processed, and then eventually, as it gets processed, it makes its way into the other side of the Golgi apparatus, where it blebs off and then is transported to the plasma membrane. And this is really what's going to make the endomembrane system. To look at it a little bit closer, here you can see here's a ribosome right here that's starting to make a protein, and that protein has this little signal sequence on the end. It's the signal sequence that's going to kind of allow this complex of messenger RNA, ribosome, and growing signal sequence to dock with a pore here in the endoplasmic reticulum, and then that polypeptide is going to be grown right into the cisternal space here of the endoplasmic reticulum itself. So now it's going to be within this compartment where it can get the right environment that it needs to fold into the right shape. Just so we can get this in our notes about the Golgi apparatus, again, what it does is this is where the final folding, the final modifications are going to be made so that those proteins, those compounds kind of know where to go. All right, so it's kind of like a shipping department. It's kind of labels things and kind of tells them where they're going to go. It's also going to start to create those transport vesicles themselves. So I think of these membrane sacs vesicles as the UPS man, right? So this is kind of like the UPS of the cell itself. Again, to look at the Golgi apparatus and the way it works, it's really this network of membranes where over here on the edges you can see the transport vesicles are starting to bleb off. and they're going to eventually make their way into the cell membrane and become part of the cell membrane. If there are any proteins or lipoproteins or compounds here in the inside of this vesicle, when it comes and forms part of, joins with the cell membrane, those things on the inside will now be released to be on the outside. It's a process we used to call exocytosis. Well, we don't used to, we still do. So here's it even closer. You can see those proteins kind of getting trapped within the transport vesicle that's being formed, right? And then that can form over here with the Golgi apparatus, like I was talking about with the bubbles. Just to recap this whole protein making process through the endomembrane system, here's our cell with a little nucleus. Within the nucleus, of course, is the DNA. That DNA is going to be transcribed into messenger RNA, which makes its way through the nuclear pore out of the cell. All right, and right there, of course, is the endoplasmic reticulum with all of its ribosomes like mad. All right, that messenger RNA is going to find a pair of those ribosomes, which then, where it gets translated into a sequence of amino acids, which of course we know as a protein. Of course, it doesn't have the right shape yet necessarily. It's through this endoplasmic reticulum, or eventually it makes its way into a transport vesicle. That transport vesicle can bleb off and then make its way to the cis side of the Golgi apparatus there, the UPS department. It's within the Golgi apparatus that polypeptide is going to get its final folding and final shipping destination labels and everything like that, make its way into a secretory vesicle. And that secretory vesicle then is going to kind of become part of the cell membrane and allow that protein to make its way into, I don't know, the bloodstream maybe, where it can go on its merry way. Just so you can have it here in picture form, right? Here's the nucleus where all the DNA is, and of course that's where the messenger RNA is going to make its way out of one of the pores here, where it can complex with some ribosome or other, make its polypeptide into the cisternal space of the endoplasmic reticulum, where over time it will eventually make its way down here where it gets blebbed off into these transport vesicles, can become part of the Golgi apparatus where it gets its final folding and final shape and final labels, whether it's going to be a transport vesicle, excuse me, a secretory vesicle that's then going to go through exocytosis to become part of the external environment, or if it becomes part of membrane, or if it becomes a lysosome. All depends on the labeling and the sorting that takes place here in the Golgi apparatus. It's a lot of information. There's a lot of parts to the cell. Cells are complex. We need to know more about the different parts of the cell. This endomembrane system is only part of the puzzle. There's more to know. Well, that is certainly enough information. Feel free to go back and watch it again if you need to see it again. Take some notes maybe, and we'll see you in class. Thanks again for checking into Mr. Ulrich's landofbiology.com. I'm Mr. Ulrich. Have a good one.